All right, so in retrospect, I did a pretty dumb thing. After I showed you this bracket, I went home and I made up a different one and I submitted that one to my pool. I picked Virginia and Illinois in the final game. Both teams, which are in the basement, they're out. So I've sunk to the bottom of my pool and the Big Ten is doing terribly, but oh well, still fun to watch. All right, so I don't really have any other announcements. Next exam is not next week, but the week after. It's on a Wednesday, so Easter's on the Sunday. Exam's Wednesday after that. So we have a little time here, but again, you can be working on your chapter 10 homework, your chapter 14 homework. Again, things to do. Where we left off time. So we'd gone through most of chapter 14, which kind of describes the process of infection of any kind of infectious disease. So we talked about some terminology. What does it mean to be a pathogen? What's an opportunistic pathogen? What are virulence factors? We kind of followed that course of infection, starting with the portal of entry, and talked about how pathogens usually need to adhere to their host, some kind of host tissue, and invade. What kind of virulence factors help them do that? And we talked about different virulence factors like toxins, exotoxins, endotoxin. Remember, endotoxin is the same thing as lipid A. It is part of the outer membrane of a gram-negative bacteria. All right, and then we kind of paused for a minute to think about, well, how do we describe the stages of infection when they're happening in a host? So this is pretty straightforward. But if you talk about, well, here's the time when the microbe or the pathogen first came into that portal of entry, it takes a little bit of time till it can kind of start reproducing itself. And to begin with, the host is not going to have any signs or symptoms. Remember, signs are things we can measure, like fever or rash or vomiting. Symptoms are things that the host feels, like headache or body aches. All right, so in that incubation period, there's no signs or symptoms. The question is that host infectious? Like, can they pass on that pathogen to other people? For most infections, no, but for some, yes. Okay, so those are the infections that they're a real problem because hosts are walking around doing their normal everyday thing. They're infectious, they're not feeling bad, but they're infectious. So of course that can happen with coronavirus, which again makes it a real problem. SARS coronavirus, which we came on the scene early 2000s, that was not the case. And that's one of the things that helped us contain that right away, because people didn't seem to be infectious unless they had symptoms. Chickenpox is another one that people are infectious during that incubation period. So that's another reason that infection spreads so easily. How long is that incubation period? It's different depending on the pathogen, very different. For something like rabies, the incubation period can be like 6 months. More often, it's going to be much less than that. Coronavirus, this is something that we've argued about. We say typically now between 2 and 14 days, most often 4 to 5 days is what we're kind of pinning that down as more. Flu, more like maybe a day. So different incubation periods. Once the pathogen is starting to reproduce a little bit more, we tend to get a little bit in the terms of signs or symptoms. That prodromal period we describe that as very mild signs or symptoms. So you kind of wake up in the morning and something's not quite right. Your throat's a little scratchy, something like that, some kind of mild symptoms. And most infections, no matter if they're really serious, like anthrax or, just a common cold, they have some kind of, kind of easing you into it kind of symptoms like that. Okay, and this is why you self-assess every day, because if you're coughing a little bit, something like that, that's something we want to pay attention to. And then it makes sense. The illness period is when you're having the most numbers of pathogen typically, and you're having the most severe symptoms. If the pathogen wins and kills the host, then that's the end of it. A lot of time our immune system starts to kind of win out, and we would get a decline, meaning a decline in the pathogen, meaning their numbers are decreasing, and convalescence is when the host doesn't have any more signs or symptoms. They may still be a little bit weakened. There may be maybe a little bit more susceptible to maybe some other infection coming in, but they're not having any more signs or symptoms. Okay, so when we take a look at some individual pathogens later on in the course, we will take care to kind of describe those stages of the disease in a little bit more detail. All right, so a pathogen needs to be able to leave that host in order to infect more hosts. Okay, so generally, it's going to leave the host in larger numbers than what it came. So we talked about what an infectious dose needs to be for a pathogen to establish an infection. It may only be 10, 100 cells or viral particles, but it's generally going to leave in much larger numbers than that, because it wants to have the best chance that it can to infect another host. Okay, so it's generally going to be shed in larger numbers. That's kind of the principle there. And it's usually going to leave in some bodily fluid. All right, so it's going to be viable for longer in the environment if it's in a nice moist place. It's not going to dry out as easily. So that's kind of the second principle there. It's leaving in bodily fluids. All right, so and again, these kind of mirror the portals of entry. A respiratory and salivary portal. This is why you've got a mask on right now, because coughing, sneezing, talking, laughing, even breathing, you are expelling, the host could be expelling microbes. In a minute here, we'll take a look at a picture of an unstifled sneeze. Okay, if you don't believe that microbes can travel on respiratory droplets, then the unstifled sneeze will kind of remind you of that. Fecal exit. So any, say, foodborne pathogen, we call that the fecal oral route. If you eat something, it makes you sick, typically going to leave by diarrhea, typically increases the motility of the bowel, right? Shedding, pathogen numbers in, huge numbers. All right, here's an example. I'll just show you briefly. If you're not familiar with the infection called cholera, this is a diarrheal disease, kind of prevalent in refugee camps, third world countries. But you typically would drink some contaminated water. So again, areas of the world where they're not treating their water source, having clean water is a problem. And this infection causes such motility of the bowel, such enormous amounts of diarrhea that patients, they get weak really quickly. They can't get up to take care of bathroom needs. So the easiest way to treat them is to just lie them on a cholera cot that has a hole in it, and they just let the diarrhea drain out of the patients. I mean, it just sounds awful. But if they can keep the patient hydrated, there's a good chance that they'll survive. They die within a day or two of dehydration if you can't keep them hydrated. So this sounds like an awful thing to experience. Another portal of exit, the urogenital tract. So sexually transmitted diseases, urine less commonly, but potentially. And of course, bleeding, again, bodily fluids, injury through a blood draw, even maybe some insect that draws blood and carries it to another host. Okay, so when that pathogen leaves the host, where does it go? Where is it maintained until it can possibly infect another host? Sometimes it will go directly to another host, and we'll talk about that transmission. But some pathogens are maintained, and we call those sites reservoirs. Okay, so places where pathogens are maintained, or where that pathogen might normally live or reproduce. All right, so 3 different reservoirs we'll describe here. Animal reservoirs. And if a pathogen is surviving and reproducing in an animal, most often, we'll say typically, the animal is not harmed. Okay, now that's not strictly true. So for example, here's a picture of a raccoon. One thing that raccoons can harbor is the rabies virus. Now, eventually rabies will kill raccoon as well, but it might be in that raccoon for 6 months or so until that happens, 9 months even. So, it kind of depends on, how long that pathogen spends there as to whether we consider it a reservoir or not. Let's see, and infections that we get from animals are called zoonoses. So, or a zoonotic infection is one that's maintained in animals, and then humans can acquire that infection when they come in close contact with an animal. So, maybe they're trapping, it's a wild animal and they're trapping it and eating it, or they come in contact with the animal feces, or the animal bites us. Okay, some kind of close contact, and then we get that infection. And there's some diseases that we pass around to each other, but we think it's, is also present in some animal population, like coronavirus, we think came from bats. There's a number of different human pathogens that bats are a reservoir. So if you have a natural aversion to bats, like most of us do, that's well-founded. Bats, again, you just don't want to come in close contact with a bat if you can avoid it. Rabies, all kinds of stuff that they can carry. Why are they such common reservoirs? Well, if you think about how they live, they cover long distances because they fly, and they live communally. So they live in, caves and, right on top of each other. They're pooping on each other as well. So they live communally, and they travel a lot. So that's why we think that bats are common reservoirs. Other examples of zoonotic infections, Lyme disease, that's the one I have down here. The white-footed mouse is a reservoir for Lyme disease, especially. Deer can be as well. Insects are not considered reservoirs. Okay, so we're talking about animals here, but not insects. We have another name for insects that we'll get to in just a minute. So, yeah, they're organisms, but they're not animals. Okay, and you can probably think of a lot of different ones. I mentioned Ebola, we think is zoonotic. We think that probably originated in bats as well. Bubonic plague, Zika, long list. Other reservoirs can certainly have human reservoirs. And again, typically for a human to be considered a reservoir, they're not actually sick. So something like whooping cough, adults can harbor a little bit of that bacterium in their upper respiratory tract and not be sick with whooping cough. But when they're around a baby that's more, has a less developed immune system, they can pass it on to the baby. So anybody that's a caregiver of a newborn baby should have a booster of the whooping cough, pertussis vaccine. And that's one that doesn't last very long. So even say you that was vaccinated when you were a kid, You should have had at least 1 or 2 boosters of that by now, and and your adult every 10 years or so. So another example of humans being a reservoir, and this is a picture from your book of some workers laying some new water pipes for a town. This was back in the 1930s. They had a breakout, or I guess that's what they would call it, of typhoid fever in that town. Typhoid fever is caused by a bacterium, Salmonella typhi. Sometimes when people get sick with typhoid fever and they recover, their gallbladder stays infected with that bacterium. And they shed it when they urinate, even though they're not sick. So they had this outbreak of typhoid in this town, and they traced it back to one of the workers that was laying this water pipe. I don't know how to say this delicately, but he would habitually urinate at his job site. Must have been urinating right in the water pipe. And like 30 people died in this town of typhoid. It has like a 10 to 15% fatality rate, right? And Typhoid Mary is probably another more common example. Her gallbladder was infected with typhoid, and she was cooking for people and getting them sick. So humans can sometimes be considered reservoirs. A reservoir can also be non-living. Okay, so something like food can sometimes be considered a reservoir. Any food that's contaminated with some kind of microbe, and we eat it and we get sick. Soil, not as many examples there, but like anthrax, something that sporulates, and those spores kind of just linger in the soil for decades or hundreds of years. The soil can be a reservoir there, or certainly water. Okay, lots of pathogens that just surviving, hanging out in water. Protozoa like Cryptosporidium, E. coli, lots of things. All right, so those are our reservoirs, right? Thinking about then how a pathogen would move from a reservoir to a host, or just from host to host. There's lots of ways we describe the transmission of that pathogen. Okay, contact transmission. It's kind of a fancy way to say the most obvious kind of transmission. You're in direct contact with somebody. So an example of direct contact: handshaking, kissing someone, sexual intercourse. Okay, you're in direct contact with another person. Indirect contact means that transmission happens via some kind of fomite. And a fomite is a non-living object. So for example, say you go into the computer, say if somebody goes into the computer lab, I don't even know if the computer labs are open right now, that has a cold, all right? They're typing their paper, wiping their nose, typing the keyboard. They leave a couple hours later, somebody else comes in, uses the same keyboard, itches their eye, oh, and now they've got a cold too. All right, so in this case, that pathogen has been passed indirectly through a fomite, which would be the keyboard. All right, so it can be a drinking glass, it could be, a tissue, lots of different things. Droplet transmission then is going to be passed by sneezing or coughing. Those large droplets that we expel kind of unintentionally sometimes. We know that pathogens can hitch a ride on those. Droplet transmission is considered less than 1 meter, right? So 1 meter, 3 feet. If you sneeze on somebody and you're closer than 3 feet, and they get ill, that's considered droplet transmission. Right. There also could be some kind of vehicle involved. So vehicle transmission, where the vehicle is considered either air, water, or food. Right. So of course there was a big argument over how coronavirus is transmitted. It's still going on, I would say. Is it airborne or is it not? Well, airborne transmission, again, air is considered the vehicle, and airborne is considered greater than 1 meter, so greater than 3 feet. So, you know, they finally came out with and said, yeah, we think that coronavirus can be airborne. You know, they're kind of revising that now, like, oh, for kids, maybe they only need to be 3 feet apart or whatever. But if it can be transmitted greater than 1 meter, it's considered airborne. We don't think that that's the primary way that coronavirus is passed. We think more often some kind of direct contact, most likely droplet transmission, probably not so much by fomites, like, you know, keeping your groceries in the garage for a few days before you get them out. That's probably very, very low risk. All right. So mostly droplet transmission. But again, infections that are airborne, it's tough. You know, something like smallpox or measles, it's more difficult to keep them from being transmitted. Water and foods sometimes are considered vehicles. Sometimes they're considered reservoirs. Again, it depends on the length of time that pathogen spends in the food or in the water. So, you know, again, I can't give you any hard and fast rules here. You know, these are just, you know, we try to describe transmission kind of as best we can. Again, this depends on length of time. Okay, if it's only, you know, in the water very briefly, then we would consider a vehicle. If it's, you know, growing and dividing there generations and generations, then probably be better to consider it a reservoir. All right, the third mode of transmission is by a vector. Okay, so vectors are living organisms, usually insects. I couldn't think of any example where we'd consider something a vector that wasn't an insect. There may be, I just couldn't think of any. Okay, so we can think of a lot of examples here. We will consider that vector to be either a biological vector or a mechanical vector. All right, so both of them are insects. A biological vector, in this case, the microbe, you know, whether it's protozoa or virus or whatever, the microbe is growing and reproducing within the insect. Okay, and then a mechanical vector, the insect is a passive carrier. Right, so let me give you some examples of each. We've talked about malaria a couple months ago. It's been already. We talked a little bit about the life cycle of malaria, how there's stages that are completed in our body. That's what makes us sick, but there's a part of that protozoa has to develop within the mosquito. All right, so for malaria, the mosquito would be considered a biological vector. Sandflies are a biological vector for a disease called leishmaniasis. And that's not something we have talked about before. Not long ago, I read it was a book about an archaeological dig, and it was talking about, you know, it was in the jungles of Peru. You've all probably heard of, you know, the Incas. They used to live up in Machu Picchu, like up on the mountaintops in Peru. And I always thought, why would they live up there? Life must have been so hard. How could they grow crops up there? It's probably cold. Why would they live up there? Well, we think now it's probably because sandflies can't survive at that high altitude. All right, so they kept more or less, more so disease free. They kept from getting leishmaniasis, which is a potentially deadly disease, by living up that high above, you know, at a certain altitude. So fun fact for today. Mechanical vector, the best example I could think of that was just a housefly. Okay, maybe a housefly that lands on, some cow manure and picks up, some E. coli or something from that cow manure, and then finds its way to the picnic and lands in the potato salad, and some microbe starts to grow in the potato salad that then gets us sick when we ingest it. So that E. coli wasn't growing or dividing in the fly in any way. It was just being carried from one place to another. Okay, so that's a mechanical vector. All right, questions, comments? All right, we'll finish up chapter 14 with some little terminology having to do with epidemiology. All right, that's pretty hot topic lately, obviously. More and more people deciding they want to go into public health. It sounds like a pretty interesting field, the study of when and how and where diseases occur. It's a lot of door-to-door asking questions, all kinds of questions of people to try to figure out, where they contracted some disease. It's a lot of statistics. It's a lot of math. So sometimes, that sounds exciting, but sometimes the day-to-day minutia of it perhaps is not. So some terminology that we use when we're trying to, describe how disease occur. Two that are related here are incidence and prevalence. Okay, the incidence is the number of new cases of some disease at some given time. And then the prevalence is the total number. So the new and the old at any same given time. So this figure in your book kind of helps understand incidence and prevalence a little bit. This is labeled as the incidence and prevalence of people with AIDS. Okay, cut out the title of this graph. It's a little bit, this graph is a little bit confusing because they have one scale on this axis on the left, that's the incidence scale, and the prevalence scale here is a little bit different. So that can be a little bit confusing. So take, for example, the incidence of AIDS. It looks like it peaked in about 1993 at about, say, 80,000. And that was new cases per, we'll say, the year 1993. At the same time, let's see, drop it down here, the prevalence, again, you got to look at this axis over here, was maybe about 150,000. Okay, so in 1993, 80,000 new cases, and then 150,000 new cases plus, so 80,000 new cases plus 70,000 people that were living with AIDS. Okay, so the incidence is always going to be smaller than the prevalence, but it doesn't, again, you got to look at the axes to understand that. So over time, the incidence has gone down. The prevalence has really skyrocketed. Interestingly, people are surviving much longer with AIDS, and that's how we can explain that. And I would say, these people are HIV positive. You're usually not considered to have AIDS unless you have, a number of opportunistic infections as well as a low T cell count. But anyway, that's how you can explain how the prevalence is so high, even though the incidence is going down. All right, so kind of an interesting graph. So you should know the difference between incidence and prevalence. Some other terms that we use to describe the occurrence of infections, not only by their frequency, but as well as their geographical distribution. All right, so these two terms, we'll start with these. I should have put them right next to each other. Endemic and epidemic. Okay, if a disease is considered endemic, it's constantly present in a population. So for instance, influenza, colds, those are diseases that are just constantly present. We know that there are. If you get an unusually high spike of some disease, then you would consider it an epidemic. All right, so I'm skipping around a little bit here. Here's a graph here that kind of tries to help illustrate that. So these are two different infections. So this one at the top is infection A, and this one down here is infection B. Okay, so infection A has a little bit higher occurrence rate than infection B. And we actually, there's some seasonal fluctuations maybe, or yearly fluctuations of that disease, but we would consider this one, it looks like it's mostly endemic, where there's some variation, but it's pretty much kind of what we normally would see. Infection B, even though we've got here numbers much below infection A, because it's much higher than what we're used to seeing, we consider that spike an epidemic. Okay, so if we normally only have 10 cases of some infection in Ottawa County, if we suddenly get 100, oh, something's going on here. We maybe would call that an epidemic, even though out of 100, a couple 100,000 people in the county really doesn't seem like a lot. Okay, so and here's another picture from your book. Normal range of something, an endemic disease, and maybe you get some new cases now and then, where an epidemic you get much higher incidence at any one time. How do we know if something's a higher incidence than normal? Well, I didn't post these, but I did post this this link right here to the CDC website. And I just did a screenshot here, and I accidentally took the screenshot that I took from last fall here, but this page is the same. This is just a partial list of notifiable diseases. And I noticed that there's a list of all of them in your book in chapter 14, so I could have just posted that. But you can go to the website. They used to publish this information weekly, and you could subscribe, and you could get these weekly publications called Mortality and Morbidity Weekly Report from the CDC. But you just click on whatever infection that you're interested in. And basically what it means to be a notifiable disease is that if you walk into a clinic somewhere and they say that you have, say, botulism, okay, they have to report that at the clinic to the CDC. Okay, so I just clicked on the data, and this is just a couple weeks ago for malaria and measles. Okay, I just kind of picked a couple. This was a little bit more interesting last fall, because you can see it compares the cumulative year to date of this year, compared it to where we were at a year ago. And that was more interesting last fall, because for measles in 2019, we had a huge problem with measles. New York City and other places, you can see, you can check by state or just, total US. This year, okay, measles, not much of a problem. Okay, we have had very little measles, and you can compare people that have not traveled with people that have, recently come in from another country. Interesting enough, with malaria in 2020 so far, we had 200 cases of malaria in the US. And of course, those would all be imported. We don't have malaria indigenously in the US. This year, we got a lot less, 40, okay? Less people traveling. So that kind of makes sense. So, we report these things, and that's how we know kind of what we normally see, and helps us understand if there's a big spike somewhere. Okay. The other terms here that I'll just come back to, if something's described as sporadic, you might see a case here or there, but it's not something that really, develops into a huge cluster. Maybe something like botulism, we get a few cases in the US every year, not really a lot of it. But something's considered pandemic if it's an epidemic on more than one continent. So, and kind of flipping back and forth here. Again, this picture is a little bit helpful here. So an epidemic, again, on more than one continent. It was about a year ago that they declared coronavirus a pandemic. And to me, that was way after we'd had a huge outbreak in Italy, we'd had, cases here in the US. So it seems like to me, we were on 3 or 4 continents before they, for like a month before they declared it a pandemic, but whatever, it's a little bit, not hard and fast rules, but kind of general guidelines, I guess. So, all right, that pretty much wraps up 14. Questions, comments? All right. All right, so we'll go ahead and move into our section on immunology. So this is chapters 15 and 16. 15 deals with the innate immune system, 16 with adaptive immune system. A lot of students I've found have some familiarity with innate immunity because you might have talked about some of those cells or processes in anatomy. 16 is mostly new for students, talking about adaptive immunity, B cells and T cells. I would encourage you, the homework assignments here are a little bit more extensive. There's a lot of animations here. So again, take your time with that. You'll get more out of it. Take your time with it. I would also encourage you to, maybe if you haven't gotten your book out very much this semester, this might be a good time. Reading the textbook, especially when we get to chapter 16, it's just seeing it one more time. I think, this gets a little bit more complicated and you really need to see it and hear it a couple of times before it will really start to sink in. So plan on spending a little bit more time here because it is a little bit more conceptually difficult than me just, throwing out definitions of pandemic and that sort of thing. So just be aware of that. And, as introduction, few slides I didn't post here, but what is the immune system? It's kind of a collection of cells and processes that protect us from invaders. I think I got this list off the CDC website at some point. These numbers are probably a little higher now because we've described some new things in the last few years, but viruses, bacteria, fungi, protozoa, parasitic worms, that's the job of our immune system, to prepare us from these things that we are around all the time. Most of the time it does a pretty good job. And also our immune system is necessary for our immune system to act in order for wounds to heal. So there's a process of little inflammation that has to take place there to repair wounds. And also, our cells are constantly dying and being replaced, and it's our immune system that cleans up. Unfortunately, sometimes our immune system doesn't work exactly like we want it to. Sometimes our immune system will act too much, and then that can cause autoimmune disease. Obviously, if it doesn't work well enough, that's going to cause problems as well. So there's a lot of checks and balances, so everything works just right. But again, that doesn't always happen. I didn't post this. I just wanted to show you, this is the sort of thing that immunologists, they love looking at things like that. Immune system is terribly complex. You can see there's all these different cells and all these signaling molecules, how the cells act on each other. So it's extremely interconnected. That's good for us because if some aspect of our immunity isn't working very well, then some other part of our immune system might be able to take over and compensate. It makes it very difficult to learn about because if I want to talk about T cells, I can't really do that very well without also talking about B cells and macrophages and dendritic cells and all these other cells. So sometimes just be aware, I might mention something and say, don't worry, we'll talk more about that later. But it's just, again, very interconnected. And we're rapidly gaining in knowledge. So the study of our immune system is one of the more recent sciences. I would say maybe it's only been 100 years or less that we understood anything about the immune system. So we still got a lot to learn, and we're learning things very, very quickly about it. Okay, so by way of kind of a roadmap of where we're going here, Your book, I kind of like the way it organizes this. It classifies our immune system into 3 lines of defense. And these are artificial constructs. There's some overlap there. I wouldn't say, oh, don't memorize whether something is a first line or second line. It's just sort of a general construct. In general, our first line of defense, we would consider external barriers and maybe, our normal microbiota. Once those things are breached, like our skin, our mucous membranes, once defenses there are breached, we would come to our second line of defense. Lots of cells to talk about there, and some processes as well. So we'll spend some time on white blood cells and other proteins like complements, interferons are protein signals, cytokines as well, the role of inflammation and fever. Okay, so that first and second line of defense, that's considered our innate immunity. In general, an innate response is one that it's the same every time we're exposed to that pathogen. So these processes, in general, there may be some exceptions here, in general don't learn. It's the same response every time. Where our third line is considered our very specific responses generated by B cells and T cells. All right, and that response is extremely specific. All right, and sometimes we call that a cell-mediated or humoral response. Typically, the T cells are considered cell-mediated, and the B cells, which make antibodies, are in our humors or in our blood. Okay, so that's just a little overview of where we're going. I like to pose this question, this thought question. Okay, so just kind of knowing what you know or knowing what I just told you. I don't know if you've ever played that game, Would You Rather? So basically in that game, they say things like, would you rather eat a snake or a salamander? And, you have to pick and explain why you would do one of those weird things. All right, so if you had to do without one of your branches of the immune system, which one would you choose? Would you rather go without innate immunity, which remember is our non-specific response, or our adaptive immunity, which is considered B and T cells that are making things like antibody, right? Usually when I ask this question in terms of a clicker question, it's pretty evenly split. Students, would choose to do without one or the other. But it turns out there is a best answer. Because of some experiments that have been done, and I don't know what pathogen they used here, but they used some mice, some knockout mice. So I just think this is sort of interesting. I didn't post this, but if you expose some mice to some pathogen, again, I don't know what they use. Normally that pathogen would, replicate to a certain amount, and given enough time, your immune system would kind of kick in and be able to clear that pathogen. All right, well, what if you took some mice here that we call them SCID mice. That means severe combined immunodeficiency mice. They are mice that are T and B cell negative. It's kind of hard to read that. Okay, so they are not able to make T cells and B cells, but they do have PMNs, that stands for polymorphonuclear leukocytes, white blood cells. And they do have another kind of white blood cell called macrophages. Those are 2 cells that are really important in the innate response. So they basically have an innate response, but not an adaptive. So you can see here that it does kind of curb the numbers, but those mice have trouble totally clearing the pathogen. Eventually, it looks like the pathogen's going to win out. And then if you take mice that are lacking innate immunity, so in other words, they don't have these 2 very important kinds of white blood cells, here the pathogen pretty much reproduces without any check at all. Okay, it turns out that those white blood cells in our innate immunity, those are necessary to call into the action of our B and T cells. All right, so really the adaptive system doesn't even ever have a chance to function because it doesn't have an innate system. So it turns out if you lack innate immunity, you would die a whole lot quicker than if you just lacked your adaptive immunity. All right, so anyway, kind of an interesting introduction to me anyway. All right, so let's start with our first line of defense. We talked a little bit about the skin as a portal of entry last class, and we said it's not a very good portal of entry because of lots of defenses that we have. Well, the skin is laid, it's a little bit more complicated than this, but we'll say it has two different layers, right? The outer epidermis and then kind of layers of cells below that. And here's a up-close scanning electron micrograph of the outer layers of that outer epidermis. You can see it looks dry and flaky because the outer layers, we have a few layers of dead cells. Okay, and those cells are continually being lost. Okay, we call that being sloughed off, or another name to say that is they undergo desquamation. They're continually falling off. Okay, so all right, desquamation. Okay, in other words, they're continually lost. Right, so you can see how this would be an advantage. Pathogens we talked about have to adhere to their target cells before they can invade deeper. If they adhere and then cells, are dropping off, then they're not going to be able to invade. Right. Where do all those dead skin cells go that we slough off? Well, they make up dust, and what eats that skin cells are dust mites. And about a third of us are allergic to the feces of dust mites. So if you have indoor allergies, you're probably allergic to dust mites feces, which again, eats your skin. If we didn't have dust mites, we'd just be walking around in, feet of our skin cells, which would not be good, but it's kind of gross to think about these little mites eating that as well. Okay, so the outer epidermis also, we'll say, has many layers of, I have trouble spelling layers, of tightly packed cells. All right, so many layers, the outer layers are dead. Deeper into the dermis, lots of defenses there. Deeper in the dermis, it contains dendritic cells, which we're going to talk more about dendritic cells shortly. They're a kind of phagocytic white blood cell that goes around patrolling for invaders. So things like your sweat glands secrete sweat. Okay, so here's your sweat gland right down here at the bottom. Sweat is salty. So we know that microbes don't often survive very well in salty environments because they dehydrate, okay, hypertonic medium. Sweat also, in addition to containing salt, sweat contains antimicrobial peptides. We won't get too specific there. As well as lysozyme. Right, so we mentioned lysozyme when we were talking about our bacteriophages and how those viruses that infect bacterial cells bring with it some lysozyme so that it can not only enter into that bacterial cell wall. Lysozyme cleaves the bonds between the sugar subunits of peptidoglycan. as well as then, burst when it's time for that, for viruses. So we'll be seeing that word lysozyme several times here. And another defense on our skin are the sebaceous glands. Okay, they produce sebum. Okay, so here is, this arrow is pointing to our sebaceous gland. You can see they're usually associated with hair follicles. So secrete sebum. This is going to lower the pH of the skin. So not only can it be a salty place because of sweat, but the pH is lower because of sebum. All right, and of course the sebum has the other function of keeping our skin kind of nice and soft and pliable. When we get too much sebum, then it can clog up those pores and actually kind of backfire and make it a more hospitable place for bacteria, and we get acne. But as long as it's in the right amount, then again, it's a good thing. All right. So in general, skin is a pretty inhospitable place. Okay, we're not very many pathogens that can establish an infection there without some kind of cut in our skin. We said that the mucous membranes were a little bit more common portal of entry. Okay, we have defenses at mucous membranes, but they're a little bit more commonly breached by pathogens. Okay, so the mucous membranes line all the different openings to our body. So reproductive tract, digestive tract, urinary tract, etc. The epithelial layer of cells, which just means the outermost layer of cells that line it, are really tightly packed to prevent pathogen entry, but the cells are alive. Okay, they're still being constantly shed, even though they're alive, but probably not as much as our skin or epidermis. And that does help carry microbes away. What kind of cells do we have lining those mucous membranes? Well, there are stem cells, deeper in deeper layers there that are going to produce a couple different kinds of cells. Goblet cells, and your book doesn't have a picture of this, so this was the best one that I could find here off the web. Here's some goblet cells. And what they do is produce mucus. Okay. It's odd to me that you spell it like that without the O, but apparently that's correct. Okay, mucous membranes with an O, and then mucus itself without it. So the goblet cells produce the mucus that helps kind of trap pathogens. There are also in that mucus some antimicrobial peptides as well as lysozymes. So there's some chemical defenses in that mucus. The mucus is slightly different depending on where in the body that it's being produced. But I've read in a couple different places now that we produce about 1 liter of mucus a day. Just enormous amount, not saliva, but mucus. So that's just an enormous amount. And you think, well, where does that all go? Apparently we swallow some of it, but okay. And then at least on the cells lining our respiratory tract, the stem cells differentiate into another kind of cell, ciliated columnar cells. Okay, and as you would expect, the cilia are attached here. Okay, so again, these are the ones lining the respiratory tract. You can see the cilia here. And of course, the job of the cilia is to kind of move out those pathogens that are trapped in the mucus and kind of help dispel them from the respiratory tract. And I mentioned this before, I think, when we first talked about, the structure of flagella and the structure of cilia. People that smoke, their cilia tends to be impaired. Not only can their cilia not move as efficiently, But eventually those stem cells stop producing columnar ciliated cells. So eventually lose the ability to make cilia at all. So in general, people that are smoke are more susceptible to respiratory infections. There aren't any peer-reviewed papers yet that talk about whether smokers are more likely to get COVID or not. I suspect that they are, but you know, I don't know that anything's been done about that. In general, their immunity, there has been studies that show people that smoke, their immunity is a little bit lower because it just kind of hampers their ability to make different aspects of our immune response, like cytokine production and the production of some other immune cells. So anyway, that's kind of interesting. Okay, also considered our first line of defense is our normal microbiota. All right, so we started talking about this last lecture. You know, we talk about how bacteria are bad all the time, but there's the bacteria that live in our body are really necessary for our health. We know for sure that they contribute to microbial antagonism. And what I mean by that is our microbiota outcompetes other more pathogenic microbes. Okay, so because we have a lot of different cells, and mostly bacteria probably, but maybe some other things too, living on our skin, you know, other more potentially bacteria that we might come in contact with have a hard time establishing themselves because our normal microbiota is using up space and using up the nutrients. So they just kind of, again, that's the strategy, they outcompete. We know that our normal microbiota somehow contributes to kind of keeping our immune responses primed. We know that they've done some really fascinating studies with mice that are raised in sterile conditions. So think about what a tough thing that would be to try to figure out, to birth mice in a sterile condition and then somehow, you know, whisk them off and raise them in conditions where there's no bacteria of any kind. But they've done that, and the mice are healthy. They don't grow as big though. And if then they are exposed to some kind of bacteria that normally wouldn't harm a regular mouse, it often kills them. Okay, they have a very, very slow immune response when they don't have any normal microbiota. So, and we know that our microbiota just improves to our overall health. They produce some vitamin B5 for us, which is necessary for glucose metabolism. Of course, folic acid is necessary for us to make nucleic acid. We get usually an adequate amount from our diet, but we get a little bit extra from our microbes in our gut as well. The microbes in our gut make a precursor of vitamin K, which is necessary for us to clot blood. So we know at least there's a lot of ways that our microbiota contribute to our health. And then if you kind of along those lines, thinking about, you know, normal secretions and processes that the way our body works, a lot of these functions are not, they have some main function that's not to act against pathogens, but they do that as well. So if you look at things like saliva, well, our saliva is to help us digest food, right? It would get caught in our digestive tract if we didn't kind of grease the wheels a little bit. But saliva also really contributes to cutting down the numbers of microbes in our mouth that would cause tooth decay. Okay, they have things like lysozyme, which we talked about, weakens the peptidoglycan of bacterial cell walls, and other antibacterial enzymes. So you produce your saliva production typically will kind of shut down when you go to sleep, right? You know, sometimes we drool or whatever, but typically it really shuts down. So practical application, it's really important to brush your teeth before you go to bed, because you want to cut down the number of microbes in there as far as you can, because they're going to overgrow overnight when they're not being bathed in saliva all the time. All right, so again, brush your teeth at night, cut down on tooth decay. People that can't produce as much saliva, there's a syndrome called Sjogren's syndrome, where people don't produce as much saliva as they should. They have a hard time with tooth decay. And again, just some other examples. Of course, stomach acid, that low pH helps us digest our food. It also is really inhibitory to foodborne pathogens. So some of them can survive passing through the stomach, we know, but that cuts down on the numbers of ones that can when that pH is so low. In your stomach, there's I think it's a protein called gastroferritin produced. This is another common theme that we see, is that our body has lots of different mechanisms to try to sequester any available iron and keep it not available for any pathogen. Often iron is kind of a limiting factor for bacteria especially. So if signals can be made for cells in our body to kind of take up any available nitrogen, we keep it from pathogens from using it. And, any just kind of movement through our digestive tract, keeps pathogens from attaching. Same thing with the movement of urine. Urine's pretty acidic, that maybe inhibits microorganisms, washes them from the urogenital tract there. Any kind of secretion, again, is just kind of flushing out mucous membranes. All right, same thing with flowing blood. If you get a cut, it's probably a good idea just to let it bleed a little bit, just kind of help wash that cut out a little bit. All right, and again, you see this iron binding proteins, binds iron. It's kind of a recurring theme that you see. All right, so that's pretty much it for our first line of defense and our innate immunity. All right, let's continue and talk about, let's spend a little time here talking about the cells. So, once our first line of defense is breached, some of those, kind of normal processes or physical barriers or our normal microbiota, then it's time to call in some cells to action and other processes. All right, so I kind of like the idea of labeling this figure ourselves. This is, from your book, and I didn't label what figure it is, but this is, again, in chapter 15. Sorry, I don't have that labeled. Let's talk about some of the important cells here that we have going on in our bloodstream. All right, so you've got some stem cell progenitor in the bone marrow. Okay, and through this process called hematopoiesis, that just means the stem cells are differentiating into more specific kinds of cells. You get these three kinds of progenitor cells. Erythroid, these are still stem cells, myeloid and lymphoid stem cells. And then from those three types, we get all the different kinds of cells in our blood. Okay, so from the erythroid cells, we get erythrocytes, or we'll call them red blood cells. All right, their function is to carry oxygen. As far as I know, they don't have any immune function, so we won't be talking about them anymore, but obviously very important. And then from those myeloid stem cells, we get platelets. Okay, tremendously important in clotting blood. That's their main function. They may have a little bit of immune function. There may be a little signaling here or there, but we're not going to focus anymore on platelets. And then we have our, make sure I label these correctly, basophils, neutrophils, and eosinophils. Okay, those are all considered granulocytes. because they contain these little dots in them. They're granules full of digestive enzymes for one thing, other things like inflammatory mediators that we'll talk more about. Okay, and you don't need to recognize these cells on site. If you want to learn how to do that, take a good histology course. I don't know a lot about that. Okay, and then a fourth kind of cell here that are also descended from the myeloid progenitor cells are monocytes. Let's spell that right. Okay, monocytes, when they leave the bloodstream and go out into the tissues, they differentiate into macrophages and dendritic cells. So again, they leave the bloodstream. We'll talk about the process of how they do that. They can differentiate into two different kinds of cells, either macrophages or dendritic cells. And then the lymphoid progenitor cells give rise to lymphocytes. And these are our B cells, our T cells, and a type of cell called natural killer cells. We usually abbreviate those NK cells. Okay, we won't talk about those too much. There's one other time that I'm going to mention them. Of course, B and T cells function mostly in adaptive immunity. The NK cells actually function more in innate immunity. Okay, so the monocytes and the lymphocytes are considered agranulocytes. That prefix A meaning without. Okay, so no tiny granules in there. And of course, all of these, the basophils, neutrophils, eosinophils, monocytes, lymphocytes, they are all considered leukocytes or white blood cells. Okay, that leuko prefix, I think means white in, I don't know, maybe Greek, Latin, I'm not sure. Okay, so leukocytes or white blood cells. So again, sometimes we got a couple different names for the same thing. All right, so that's a little overview of the cells that we're going to be talking about. Let's talk first about our granulocytes, and in particularly the neutrophils, because there are so many more neutrophils than basophils or eosinophils. Another name for the neutrophils, they're also called PMNs. And I mentioned that before briefly. That stands for polymorphonuclear leukocytes. Okay, now that name makes sense. If you look at the picture here, here's the nucleus in the center of the cell. It's got many lobes. Okay, polymorph, many shaped, or many lobes of its nucleus. Okay, and it's a white blood cell. So, so technically, basophils and eosinophils are polymorphonuclear leukocytes as well. But because there's so many more neutrophils than there are basophils or eosinophils, most of the time when we're talking about PMNs, we use that as synonymous for neutrophils. Okay, so technically a neutrophil is included in that group, but usually we consider them the same thing. Okay, about 50 to 60% of the white blood cells in circulation, so that means in the bloodstream, are neutrophils, right? So they outnumber the other ones by quite a bit. They're considered highly motile. Okay, they're not moving by flagella, they're moving by amoeboid action. All right, so they kind of, make their way through the tissues. Of course, they can be flowing in the bloodstream as well. They're considered first responders. All right, so think about neutrophils as kind of the ambulance, the first help on the scene of some injury or infection. They're usually the first cell there to try to clean it up. They're pretty short-lived. I read different things, but typically they're considered to survive less than 24 hours. Okay, so we're constantly making neutrophils in, again, in our bone marrow and replacing them. We'll talk about why that's not really a problem. We think about how they work. Usually when they move into the tissues out of the bloodstream, it's only maybe a couple hours that they live before they stop functioning. Okay, so what do they actually do? We think that there's 3 different ways that neutrophils help kill invaders. Okay, phagocytosis is a primary role, and we're going to look more at the stages of phagocytosis a little bit later. We know that they act by degranulation, so they're releasing these kind of digestive enzymes, as well as mediators that will increase inflammation. We always think about inflammation as being a bad thing, but on a short-term thing, it's going to help bring more cells to the site of where there's an infection. So short-term, it's a good thing. We'll talk more about that as well. And the one method of killing that's unique to neutrophils that was only described about maybe 10 years ago is the formation of nets. So that stands for neutrophil extracellular traps. Okay, so this picture that I came across, you can't see the neutrophil in this picture. This rod-shaped bacterium here is a Klebsiella pneumoniae in the lung of a mouse. And the neutrophil has basically blown itself up. It expunges its DNA. If you've ever extracted DNA from some cells, the DNA is quite sticky. All right, so it kind of expunges its DNA. Maybe we could think about it as like throwing a hand grenade or something in order to mobilize some invader. So basically, the neutrophils sometimes will sacrifice themselves in order to keep invaders from, moving on or, invading deeper into tissue. Okay, so that's kind of a cool picture there. I don't know how they got that. But again, that's only neutrophils that we think can do that. Here's kind of another interesting picture of a neutrophil acting to phagocytize some kind of, I think this was supposed to be anthrax, yeah. So these rods here are anthrax. The thing that puzzles me about this picture is that neutrophils, they're eukaryotic cells, they should be way, 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 way bigger than any bacterium. So this neutrophil is like, barely big enough to phagocytize it. So it seems kind of odd, but anyway, again, kind of an interesting picture. Okay, and just, I know our time is short here, but the other 2 types of granulocytes are the eosinophils and basophils. I put them together here on one screen because their function is quite overlapping, and we don't have very many of either one. We know that both of them are primarily acting by secreting the granules, and they're generally not phagocytic. There may be some exceptions to that, but generally they're not. They don't use the nets. The eosinophils, when you have an elevated eosinophil count in your white blood cell count, that probably tells us that the host is fighting some kind of eukaryotic pathogen. Helminth is a eukaryotic worm, like a fluke or something like that. So that, or it could mean an allergic reaction. But sometimes we get a hint there. again, the basophils, there's a lot of overlapping function here, also plays a role in targeting those eukaryotic pathogens, okay? Also a role in allergic reactions as well when they degranulate. All right, we will pick up there on Wednesday. Enjoy the day.